Good morning, Mr. Vaibhav.
Hi, good morning.
Good morning. So we are going to start, Kalpana? Or should we wait for 1 or 2 minutes?
Sir, just a moment. I was waiting for director sir to be in.
Okay.
Yeah.
Ma'am, it's 11 a.m. so the live stream has started on YouTube.
Okay, so a very good morning, everyone. Speaker of the session, Mr. Vaibhav Kumar, Dr. Rajendra Dhaka, Chair INYAS, and Dr. Sandeep Arora, Director, Amity Institute of Pharmacy, and dear participants all around the globe. I am Dr. Kalpana Nagpal, and on behalf of Amity Institute of Pharmacy and NCR Chapter INYAS, I welcome you all for this very interesting session on the key aspects of pre-formulation studies. So as students, you might be busy in running experiments, analyzing the data, reading, writing, acquiring new knowledge, and might be aware about developing new skills so as to make yourself future ready and get a suitable job in future in pharmaceutical industry. But we rarely think about how to connect with someone to get that quality knowledge, exposure, and guidance beyond our peers or teachers, especially the real examples that industry people come across with and deal with them to launch a product in the market. So the idea of this expert lecture here is first to analyze what is the demand from the industry, what are they pursuing, how to meet that requirement of the industry. So this lecture is actually an attempt or as an effort to get that exposure and knowledge directly from the industry expert and let us learn in a more effective way towards updating our existing knowledge about pre-formulation study and acquire new knowledge in this session. I hope this session will brew some interesting interactions in this direction. That's why we are studying pre-formulation, why we are reading that or studying that. What are the tests which are exactly being conducted under the pre-formulation aspect studies, including bulk characterization, the stability of the drugs, their solubility studies as an overview. So may I now request Dr. Dhaka, Chair INYAS, to brief us about the origin of this initiative and its relevance. Dr. Dhaka, please.
Thank you, Kalpana. Good morning, everyone. So on behalf of INYAS and the organizing team, it is my pleasure to welcome our invited speaker of this special session, Mr. Vaibhav Kumar. I also welcome all of you who have joined through the Zoom platform, as well as some of you are watching through INYAS YouTube channel. As Mr. Vaibhav Kumar is also, I think, first time on the INYAS platform, let me start by giving you an overview of INYAS and its mandate. And I'm sure all students are also probably first time on this INYAS platform. So INYAS stands for Indian National Young Academy of Sciences, which was established and founded by INSA in late 2014, and currently the only recognized academy for young scientists in India. Currently in INYAS, there are 100 plus current members and 45 alumni members from all over India and from various disciplines. So let me tell you about our vision. Our vision is to promote science education and outreach, networking, and support for young scientists, as well as build scientific temperament across the country. We organize many outreach activities like science camps at school level, awareness workshops, and skill development and competition events for various levels, like for college students, for PhD students, as well as for postdocs. We also conduct various surveys on different scientific issues faced by young researchers, young students, and then we analyze those responses and publish them as white paper. And not only that, we also communicate those results and those responses to different policymakers for a positive change in the country. INYAS organizes various conferences to have more networking among early career researchers, aiming to have more interdisciplinary research for future collaboration. So I can tell you that INYAS is a great platform for young scientists to learn from each other, and more importantly, to work together and contribute back to society in different ways. And in these directions, we have taken many new initiatives in recent past. And one of them is to connect between industry and academia, as Dr. Kalpana also mentioned in the beginning. And we all understand and know that industries are playing a big role in the growth of society. So under this, we understand that pharmacy is a very competitive and diverse professional field and holds well for healthcare industry. We were able to overcome the pandemic situation of COVID-19, as we all know, only because of the advancements in our pharmaceutical sector research. From knowledge perspective, this subject is of great scope in industry as well as academia. So to make you all aware about the recent trends in profession, we came up with this subject expert lecture. As Kalpana mentioned, the key aspects of pre-formulation studies. The main target is to give you all the adequate exposure, especially those who are among you in your undergraduate as well as at postgraduate stages. The knowledge about the pre-formulation studies, I'm sure, will help you screening lead candidates based on the understanding of their physical, chemical, and biopharmaceutical properties. This lecture will make you aware about the evaluation of medical compounds before actually they are being converted into the dosage in the form of tablet, capsule, injection, syrup, and etc. As conveyed to me, the topic is the inherent part of pharmacy curriculum and very relevant to acquire knowledge if you are looking for good job opportunities in academia as well as industry in this sector. So I request all participants, all particularly to students, to interact with our esteemed speaker and gain the knowledge in this subject. I'm sure this interaction will benefit all of you for your future. Thank you all.
Thank you so much, Dr. Dhaka. It's indeed my honor and privilege to introduce the speaker of today's session to our audience. Before that, Dr. Dhaka, I just wish to show you whether you have able to see or not. The students are present in my classroom also. So,
oh, that's really great. I was not knowing that you have full class of students there. That's really
yes.
great.
Yeah.
And
So,
also students from various places through Zoom and YouTube channels.
yes. Yeah. So I just wish to mention that.
Thank you.
Okay. So regarding the resource person is Mr. Vaibhav Kumar. Mr. Vaibhav has a rich experience of teaching as well as industry, and especially the lead roles. He has a total industrial experience of about even more than 15 years. He has also worked with Sun Pharmaceutical Industries Limited, erstwhile the Renbexi Laboratories Limited, from January 2007 to October 2020. So I think it's a long time. I mean, at present, he's heading Center of Excellence Labs in Milan. His expertise is in risk assessment, method development, method validations, method transfers of analytical method for determination of trace level nitrosamine impurities in drug substances, as well as the drug products. So it's our pleasure to have Mr. Vaibhav Kumar here with us for this particular lecture. I'm really grateful to you, sir, for accepting my request to share your learnings and experiences with the young generation, even for the working day today for you. Thank you so much, sir. Now, without wasting much time, may I request Mr. Vaibhav Kumar to please share his words of wisdom. We all are eager to listen to you, sir. The floor is all yours now.
Okay, thank you, Dr. Kalpana, and thank you, Dr. Dhaka, for a brief introduction of INYAS India. So it's my pleasure to speak in front of all of you. And as I am away from academics since last 15 years, so I am away from reading. But still in industry, I am learning a lot in industrial aspects, various various studies we are doing in industry in line with the expectation of regulatory agencies. So the purpose of a pharma industry to make a good, safe and cost-effective formulation for the patient safety. So patient safety is the prime requirement for the pharma industry, although business is also requirement. So if business is not there, so pharma company will not be exist. So the business of a pharma company majorly depends on the pre-formulation study, as it is the base how we formulate a formulation, how safe formulation we are making. So all pharma industry is reliant on pre-formulation studies. Although it is related to the pharmaceutics part, formulation part, but in pre-formulation studies, we have to learn various aspects, including analytical research and other chemical research also, it is also involved. So I take it as an opportunity to explain my experiences with all of you on pre-formulation studies. So here we will understand what is the pre-formulation studies, what are the goals and objectives of pre-formulation studies, and what are the physicochemical parameters involved. And we need to understand what physicochemical parameters which are affecting the release of a formulation, which affect the safety of a formulation. We understand various pharmaceutical excipients and their interactions, and as a case study we will take as a extended release formulation, as it is a complicated formulation, and this formulation will release slowly into our body. And the importance of extended release formulation is to reduce the number of doses in a day. Suppose we are taking 3 times a day. If we are formulating extended release formulation, we have to take only 1 tablet per day. So it is the importance of extended release formulation. To formulate extended release formulation, we have to use various excipients. We have to study various physicochemical parameters of the API as well as flow properties, or all these aspects we have to study. So coming to pre-formulation studies, it is the base of research and development process in which we have to study the physicochemical properties of a new drug substance and their interaction with excipients in order to develop stable, safe and effective dosage form. And major objective of goals and objective of pre-formulation studies or a formulation development, we have to develop an elegant dosage form which is safe, effective and affordable to the patient. In this, we have to establish various physicochemical parameters of a new drug substance, various physical characteristics such as particle shape, particle size, flow properties, and to establish the kinetic rate profile, how drug release inside the body, and to establish the compatibility with common pharmaceutical excipients which we are using to formulate the formulation. Coming to the principal areas of pre-formulation studies, I will cover in this these 3 aspects: bulk characterization, solubility studies, stability studies. In bulk characterization, we have different properties of a API. API can be crystalline, can be amorphous. Various polymorphic forms are available for API. These polymorphic forms have their importance. Hygroscopicity also one factor which is very important to be taken into consideration while formulating a formulation. Particle characterization, powder flow. If we come about the solubility studies, solubility, you know, if drug is soluble, it can absorb into the body, and if it can absorb, it will show its response. So solubility is one of the important parameter we have to taken into consideration while formulating a formulation. And stability also very important, because if we try to formulate a formulation and the formulation is not stable, it will not be looking elegant to the patient. It will not be safe to the patient. Various impurities can be formed while during shelf life of a formulation. So if a tablet or if any formulation is available in the market, it has to be kept at particular storage condition into the chemist shop. And if various stability related parameters we are not taken into consideration while formulation, it will be very dangerous for the patient, because unavoidable impurities can also form during shelf life, and those impurities can be harmful for the patients. So as a formulator, as a human being, since these medicines are consumed by our loved ones, so it is our prime responsibility to develop a formulation which is safe for the patient. Talking about the bulk characterization, we study crystallinity, polymorphism, hygroscopicity, fine particle characterization, and powder flow. So if we talk about crystallinity is like it is the inherent character of a API, in which the description of a crystal, outer appearance of the crystal is visible for the API. So if we observe the API particles into the microscope, we can see the different shapes of the crystal formed for the API. And there is significant importance for these different type of shapes of the crystals. Internal structure in this crystalline and amorphous, there is significant difference between crystalline and amorphous. In crystalline, there is fixed internal structure, whereas amorphous, they do not have fixed internal structure. Usually crystalline forms are more stable and amorphous forms are less stable due to high thermodynamic energy. And solubility also, if we see, if we want to have a more soluble API, we can go for amorphous form, and if we want to have low solubility of API, we can go for various crystalline form. Since crystalline form are more stable, they have fixed internal structure, so they have less tendency to change its form from during storage. And while amorphous, they tend to revert to more stable form during storage. So amorphous, as such, it is less stable. In storage, it tends to revert back to the more stable form. In polymorphism, we can the API characters or crystalline form have more than one form. If more than one crystalline form is available in API, then we call it as a polymorph. For example, one API chloramphenicol palmitate, it exists in 3 polymorphic forms. So all polymorphic forms have their different physicochemical parameters. They can have different solubilities. They can have different particle size and shape. So when we select the polymorphic form, it depends on the intellectual property function. As we are working in a generic pharma companies, so we have to take consideration of IPR also. If an innovator is formulating a drug with one polymorphic form, and if he has a patent, then we cannot use the same polymorphic form. We have to develop a formulation by taking other type of polymorphic form. So there is very significant importance of polymorph. Such a stable polymorph represents lowest energy state and the highest melting point and least solubility. During process of crystallization, we have some compounds which has tendency to trap solvent molecules. So during crystallization, this molecular complex has incorporated the crystallization solvent molecules into specific sites within the crystal lattice and has stoichiometric number of solvent molecule complexed. So you heard about anhydrous, hemihydrate, monohydrate, dihydrate. So if any compound will entrap water and make complex is called hydrates, and if the solvent is other than water, it is called as solvates. So if one mole of compound mixed with 0 mole of water is called anhydrous. In this, water is not present. If 1 mole of compound mixed with half mole of water, it will be hemihydrate. If 1 mole of compound mixed with 1 mole of water, it is monohydrate, and 2 moles of water is dihydrate. Similarly, you can say pentahydrate, hexahydrate. These APIs are also available. So they have significant importance. Generally, the amorphous form of the drug has greater aqueous solubility than the hydrate forms. This is because the hydrate is already in equilibrium with water and therefore have less demand of water. For example, anhydrous form of theophylline and ampicillin have higher aqueous solubility than hydrate forms. If we say non-aqueous solvates, they have greater aqueous solubility than non-solvates. For example, chloroform solvates of grisofulvin are more water soluble than their non-solvate forms. Let's come to on hygroscopicity. Many pharmaceutical material have tendency to absorb atmospheric moisture, especially water soluble salt forms. In this, if compound is hygroscopic, it will affect our formulation, it will affect our dosage form design. So we need to understand the hygroscopicity while formulating a formulation, and the equilibrium moisture content depends on the atmospheric humidity. If atmosphere is more humid, the chances of entrapment of water into the compound is more. It depends on temperature, it depends on surface area of the particle. If surface area of particle is more, if the form is amorphous, there are great chances of entrapment of water. Exposure time of a substance and mechanism of moisture uptake, how it is entrapping the moisture. Some compounds are very deliquescent. They absorb sufficient amount of moisture and dissolve completely in it. For example, calcium chloride, anhydrous calcium chloride, it has tendency to absorb moisture and completely dissolve in it. It liquefies very soon. For example, ammonium formate, ammonium acetate also. If we expose it into the air, they will convert into the liquid form. And what is the significance of hygroscopicity? Hygroscopicity is to decide the special handling procedure. If material is hygroscopic, we have to take care while handling these kind of materials with respect to time. So if we give more time while weighing, if we will give more time while handling this material. Hello, sorry. Hello. So it will take more time. If we take more time for handling, it will absorb more moisture. So it will be dependent on the time. It will decide the storage condition. So if material is hygroscopic, we have to store it well in closed glass containers. Special type of packaging material are required. Special type of desiccants are required to avoid the entrapment of the moisture. Moisture level in the powder sample may affect the flowability and compactability of the substance, which are the most important parameter while tabulating and capsule filling. Moisture can degrade some material, so that if moisture entrap into the drug substance, it can increase the impurity level in the API, which may be harmful for the safety. After absorption of moisture, if hydrates are formed, then solubility of that powder may also change, and which will ultimately affect the dissolution profile of a compound. Comes to powder flow. Powder flow property depends on particle size, density, particle shape, and electrostatic charge, and absorbed moisture that may arise from processing or formulation. A free-flowing powder may become cohesive during development. So this problem may be solved by any of the following ways. If we try to compact a powder, if we will start formulating by taking a free-flowing powder, during the process it may entrap moisture, and to avoid these things we have to granulate them. We have to by densification via slugging, we can break the particles by filling special auger feed equipments in case of powder, by changing the formulations. Let's come on solubility. Solubility is very important part in order to release the formulation inside the body, and to understand the compound, how much it is soluble, we have to establish the equilibrium solubility of a substance. The drug is dispensed in a solvent, then the suspension is agitated at the constant temperature. It is the procedure to determine the solubility, and solvent, we have to establish solubility in various solvents like 0.9% NaCl at room temperature, 0.01 molar HCl, 0.1 molar HCl, 0.1 molar NaOH, and different type of pH buffers at 37 degrees Celsius. So the equilibrium solubility concentration we have to check with all these solvents, and in order to understand what is the solubility of a compound. So what is the significance of solubility? Solubility, as we are taking medicine, if medicine contains active pharmaceutical which is not soluble, it will not absorb into the body. So the solubility is very important role, plays a very important role. And a drug for oral administrative capabilities should be examined for solubility in an isotonic saline solution and different pH. And this solubility data may provide the dissolution profile of the drug. Solubility in various mediums is useful in developing suspension or solution or toxicological and pharmacological studies. And solubility studies identify those drugs with a potential for bioavailability problems. So if substance is more soluble, it will be more bioavailable. Drug having limited solubility, 7% in GIT fluids, often exhibit poor and erratic absorption unless dosage forms are tailored for those drugs. We have to modify the dosage form in such cases to enhance the solubility of a active pharmaceutical. Coming to stability, stability is very important. We have to take care of various impurity formed during shelf life of a formulation. We have to establish the proper storage area of the compound. We have to establish various packaging materials for the dispensing of the formulation. So stability is very important. We need to know the stability study in solution state. We need to know the stability study in solid API state also. The stability study in toxicological formulation, a new drug is administered to animals through oral route, either by mixing the drug into the feed. So if the feed is interacting with the drug, it may, if it is not stable, it may convert into different another form, and that effect will not come during the study. In the form of suspension in aqueous vehicles also, we have to take care. Stability of a new drug may depend on pH of ionic strength, co-solvent, light, temperature, and if we say stability, we have to check the stability of a compound across different pH range, starting from acidic to the basic, because we will take the medicine, it will travel from stomach to small intestine to large intestine, and different pH range will be there. So by interacting with different pH of biological fluids, it may not stable and may decay, may form different impurities. So we need to study stability studies for a different compound to be formulated across the different pH range solvents at very high temperature also. So these are the extreme conditions which we check the stability of a compound. So if compound is stable at these extreme conditions, that will be safe for our body. That will not degrade, that will not decay. One property is co-solvent. Co-solvent, in this case, some drugs are not sufficiently soluble to give concentration of analytical sensitivity. So co-solvents may be used to increase the solubility. Some compounds are light sensitive. So to avoid decay of light sensitive compound, we have to keep the solution in clear glass, amber colored glass ampoules or yellow colored glass containers. Or if we want to study these things, we have 4 different parameters. We can take a control sample as stored in cardboard package and wrapped in the aluminum foil. That will be considered as control, and we will keep substance in clear glass ampoule, amber colored and yellow green colored glass container. So we will compare all decay in these different containers. So if it is light sensitive in clear glass ampoule, it will be degrading more. So now the stability studies are carried out by using these procedures. We use API for formulation of a drug formulation, and to study the stability of API, we need to check the storage condition, stability of a API in these storage conditions. We keep API at refrigerated condition, room temperature, ambient temperature, ambient humidity, 3775 RH, and extreme temperature also we check the stability of the substance. So in India we have different temperature zones. In north of India we have extreme temperatures from like 40 degree in summers and 0 degree in winters. So we need to check the stability of a substance in all the temperature ranges. Drug excipient stability profile also need to be established. Hypothetical dosage forms are prepared with various excipients and are exposed to various conditions to study the interaction of drugs and excipients. So if we talk about the summary of all which we discussed, the property which impact what part of the formulation, what part of the parameter. So melting point and boiling point will affect the purity determination or stability of a compound. Solubility will affect the assay, dissolution profile, bioavailability, and stability. pKa is having impact on stability and bioavailability. Partition coefficient has impact on stability, bioavailability. Polymorphism, different MOFs have impact on stability. Few forms are more stable, few forms are less stable. So polymorphism may impact the stability, solubility, and bioavailability. Particle size, shape, and surface area may affect the dissolution rate and stability studies. Interaction with excipients will affect the dosage form design, stability, and bioavailability. And flowability, it impacts the formulation development. Hygroscopicity impacts the stability. Let's come on pharmaceutical excipients, which are the excipients which we use in formulating a dosage form. These are the ingredients which along with API make up the dosage form. Any active pharmaceutical ingredient cannot be administered as such. We have to make a dosage form in order to give ease to the patient. Excipients are generally added along with the active pharmaceutical ingredients in order to protect, support, or enhance stability of the formulation, bulk up the formulation in case of potent drugs, for assisting in the formulation of an accurate dosage form. Some drugs are very potent, have low claim, like 1 mg, 0.5 mg. So that low claim drugs, we have to make a formulation of an appropriate size so that patient can take easily as a solid dosage form. These excipients help improve bioavailability of active drug substances, enhance the stability and solubility, enhance the overall safety and effectiveness of the formulation during its storage.
Sir, please pardon. Pharmaceutical excipients, which are the pharmaceutical excipients which we are using in formulating a formulation. So it is example of solid dosage forms. We can use diluents. Its function is to provide the bulk to the dosage form, and various diluents are being used in the formulation, such as lactose, starch, dextrose, MCC, and binders and adhesive. They have the function to impart cohesive qualities to the powdered material. So when we are formulating a tablet, we have to use some binders or adhesive, and these binders and adhesives will improve the flow property of a compound, will provide the cohesiveness of the powdered material, will help in the formulation of granules. So for example, binders have examples like acacia, gelatin, starch paste, PVP, CMC, carboxymethyl cellulose, etc. Regarding lubricants, lubricant has function to reduce interparticular friction, prevent adhesion of the tablet material to dyes and punches. So lubricant are required, like when granules are traveling from hopper to the punches. If lubricants are not there, they can stuck. They can stick to the walls of the hopper. They can stick to the machine, and they can stick to the dies and punches. So in order to avoid these kind of problems, we have to use lubricants, and they works on the principle to interpose a film of low shear strength that interface between tabulating mass and die wall. For examples of lubricants are talc, magnesium stearate, polyethylene glycol, stearic acid, calcium stearate, etc. These are the example of glidants. Glidants usually improve the flow characteristic of the powder material. Colloidal silicon dioxide, asbestos-free starch, cornstarch can be used as glidant. They are added in dry state prior to the compression to reduce the friction between the particles. Disintegrants are like starches, cellulose, cross-linked polymers. So they will facilitate to break or disintegrate the tablet after administration. It is very important, because if tablet is ingested into the body, it should be disintegrated for proper dissolution and to get the proper effect. Sometimes we have to formulate a formulation which is which is easily like dispersible tablets, in which we have to use super disintegrants. Super disintegrants improve the disintegrant efficacy. It will function by drawing water into the tablet and swelling it and causing the tablet to burst apart. So example of super disintegrants are prospovidone, sodium starch glycolate. So these are all theory. These you can read from literature. We will talk about the case study. Let us take an example of extended release tablet, formulation of extended release tablet containing felodipine. So when we want to formulate an extended release formulation, we have to understand various physicochemical parameters of the drug substance, as well as we have to select appropriate excipients in order to get the desirable release of the tablet. So for all orally administered formulation, optimization of pre-formulation and formulation parameters are required to assess the performance of the dosage form. Felodipine is class 2 drug as per BCS classification. Class 2 means it has low water solubility and high permeability. So its property is its water solubility is low, but it is highly permeable in the biological membranes. Ingredients with low water solubility cause formulation problems due to their reduced dissolution rate in aqueous media and therefore in the gastrointestinal tract. These characteristics results in low absorption of the therapeutically active drug substance, thus having reduced bioavailability after oral administration. A formulation, when we are talking about a development of a formulation, we should refer to an innovator product. In this case, innovator product is Plendil 10 mg prolonged release tablet. It is from AstraZeneca, and its formulation contain these different kind of excipients: HPMC, lactose monohydrate. Lactose monohydrate worked as filler. Microcrystalline cellulose worked as filler. Polyethylene glycol worked as lubricant. Sodium lauryl sulfate worked as solubilizer. Since felodipine is low water soluble, we have to use solubilizer also into the formulation to enhance the solubility. Propyl gallate used as antioxidant. Magnesium stearate used as lubricating agent, and polyvinyl pyrrolidone used as binder. If we see the felodipine, in this study we used two different forms of felodipine. One is macrocrystalline form, another one is microcrystalline form. Macrocrystalline form is bright yellow homogeneous crystal, and microcrystalline is green colored homogeneous crystal. So to study the physicochemical property, we have to do FTIR study for two felodipine forms, and we have to check the FTIR data with our reference product. So our two felodipine forms have approximately the same spectra as of the innovator product. The red one is innovator, and this blue and green one is our two different forms of felodipine. So these are the FTIR spectra from 400 to 4000 to 3000, 3000 to 2000, 2000 to 1000, and 1000 to 400 wave numbers. To study the melting point of the API, we use DSC, differential scanning calorimetry. So in this microcrystalline felodipine, macrocrystalline felodipine and reference material are subjected for this study, and we found that the reference material has lower melting point compared to our forms of phenoldiene. The melting point of reference material is 143.9, while microcrystalline form has 146.8 and macrocrystalline form has 148.51. The lower melting point of microcrystalline phenoldiene form is due to increased surface area. Because particle size is reduced, so surface area is increased, and with increase in surface area, melting point reduced. If we check the physical property of the API, we need to use the microscope. With the optical microscope, we can see the clear difference between macro and microcrystalline phenoldiene. Macrocrystalline phenoldiene are larger in size. They have clear visible crystals, and while in microcrystalline phenoldiene, we can see the shape of the particle and how it looks. So this is the clear difference between macro and micro with the help of optical microscopy. Determination of particle size. How we determine the particle size? We can determine by direct microscopic examination using computerized image, or by with specialized software, and by sieving and sifting method. So if we see the macrocrystalline form and microcrystalline form, the particle size is average particle size is around 3000 micron, and for microcrystalline it is around 30 micron. So there is significant difference between the particle size, and particle surface area also has significant difference. If we see the bulk density and tab density, the microcrystalline phenoldiene have low density compared to macrocrystalline phenoldiene. This is 0.75 is bulk density and 0.83 is tab density of microcrystalline phenoldiene, and 0.29 and 0.34 is bulk density and tab density of microcrystalline. If we talk about the extended release formulation, means the formulation when ingested into the body, it will release with slowly with time. So it is the most rate limiting factor which is affecting the formulation. So to study this release profile, we have to perform in vitro dissolution. In vitro dissolution profile of phenoldiene obtained will be compared with the reference product. Reference product is innovator tablet. So in vitro dissolution studies were performed using USP type 2 dissolution apparatus, which is paddle, and the dissolution media which we used as 0.4% citrimide and 1% sodium lauryl sulfate in phosphate buffer, pH 6.5. Amount of phenoldiene dissolved at different time interval, 2 hours, 4 hours, and 12 hours was determined by using 6 tablets using UV method, ultraviolet spectroscopy. So if we check the dissolution data which we obtained for different type of formulations, in this 9 different type of formulations were formulated by varying the HPMC content, HPMC 5, 10, 50 and 100. It is based on the complexity strength of the HPMC. Microcrystalline cellulose also we can vary to check the effect on dissolution. So these are the dissolution profile for all the 9 formulations in 0.1% sodium lauryl sulfate and 0.4% citrimide. So in this case, when we want to compare our dissolution profile of our product with the innovator product, we have to determine the difference factor and similarity factor. Difference factor is F1, similarity factor is F2. So difference factor is like average percent difference over all the time points in the amount of test brand dissolved as compared to the reference material, reference brand. So if this value is close to 0, it will be identical profile, and if value is increased, it will be the profile is different. So in this case, some values are 11, some values are 92. So as value increases from 0, difference between the formulation will increase. So this F1 is calculated with reference with F1 formulation, reference with F2 formulation in this media. If we talk about F2 is similarity factor, and the similarity factor is the simple measure of the comparison of two dissolution profile. If value is near to 100, it will be like similar. If value is lower to the 100, it will be non-similar. So the value of F2 should be more than 50 and should be near to 100 if we say that our profile is matching. So as a generic company, if we are formulating a formulation, the release profile should match with the innovator. To check whether our profile is matching with the innovator, we are using these tools by calculating F1 and F2. So with all 9 formulations, if we can see, formulation 8 has low value of F1, which is like more similar to innovator, and 62.91 value of F2, which is more than 50. So this formulation could be the best among 9. If we take inferences from these 9 dissolution profiles, that increasing the amount of cellulose polymer HPMC K100, which possesses the highest viscosity degree in the composition of experimental formulation, that lead to delay in the release of phenolidipine, correlating with the added percentage in all investigated dissolution media. And when 2 HPMC polymer are combined in the formulation, it was found that the presence of larger proportion of higher polymerization degree like HPMC 100 or the viscosity polymer led to decrease in the phenolidipine release rate. The release in higher surfactant content dissolution media led to an increase in the release amount respecting the above particularity. The use of surfactant was for increasing the solubility profile. The release was lower in CTAB than 0.1% sodium lauryl sulfate. Sodium lauryl sulfate is surfactant which enhance the solubility of the phenolidipine. That's why release was higher compared to CTAB. If we talk about the tablet containing 15% of high density HPMC, it will lead to 94% of the dissolution in 2 hours. Let's see in this. So that F1 in this release was more than 100% in 2 hours. So this formulation is not an extended release formulation. Our purpose is to make an extended release formulation, and then that the formulation should release slowly with time. If formulation is being released in less than 2 hours, that purpose is not met. So we cannot use this HPMC 100 in the same medium. F2 with a concentration of 30% HPMC 100 showed 8.7% of dissolution in 2 hours and 77.5% dissolution in 12 hours, which is very, very comparable to innovator product. In this innovator, 8.35% in 2 hours and 87.08% in 12 hours. Two different formulations were formulated by selecting the different particle sizes, particle diameters. So if we see the phalodipine microcrystalline 5 mg, phalodipine microcrystalline 5 mg in formulation 11, formulation 10 macro, formulation 11 micro. Rest of the excipients kept same. In this, if you see the dissolution profile, in media 1% sodium lauryl sulfate, the phalodipine dissolution differed significantly between F10 and F11, while in case of CTAB, the profile of F10 and F11 are similar. That if you see the F10 and F11 in 1% sodium lauryl sulfate media, F1 value is 49.86. That means difference is more, and similarity factor is less than 50. That means difference is more. In CTAB media, difference factor is 20, which is less than 49, means it is much similar, but still the purpose is not met. Although the similarity factor F2 is more than 50, but it is not releasing 100% release. So the release is less than 60%. In this case, we need to change the formulation. So if we talk about the tabulating force, we formulated two different formulation with different tabulating forces, 45 Newton and 90 Newton, keeping the all ingredients same. And dissolution was performed in 1% sodium lauryl sulfate and 0.4% CTAB. And if we see the difference factor between F12 and F13 is 7.13 in 1% sodium lauryl sulfate and 9.34 in 0.4 CTAB. So it is less than 10. It is much closer to 0. Profile are matching. Similarity factor F2 also more than 50. So these formulations, we are just achieving our target. Increase in the compression force or decrease in the compression force does not have any impact. So compression force can be kept as 45 Newton as well as 90 Newton. That is not impacting much on the dissolution profile. If we check the dissolution profile of quantity used as lactose monohydrate. So in this, we see lactose monohydrate is 5 mg in formulation 14 and 16.5 mg in formulation 15. So in this case, it is clearly visible, this orange dissolution profile. This is much faster compared to all other profiles. In this case, formulation 15, high lactose content was used, and the higher lactose content led to the marked enhancement in the release of phalodipine from HPMC matrix and cause increased difference in the dissolution in two different medias. So this phenomenon can be attributed to the dissolution in the polymer matrix, increasing of the pore forming excipients content and thus extending the content surface between the phalodipine particle inside the matrix and the solvent. Not changing. Coming to formulation 16 and 17, in this different proportion of microcrystalline cellulose and lactose monohydrate was used. If you see lactose monohydrate 14 mg and 21 mg in formulation 16 and 17, and microcrystalline cellulose is 12 mg and 5 mg. Rest other excipients kept same. So in this case, if you see the orange, it releases more than 100% in 2 hours. So this is not an extended release formulation. We cannot use this formulation further. by increasing the amount of lactose by 7% and decreasing the amount of microcrystalline cellulose by 10%, the phenoldiphine availability was enhanced with this media, sodium lauryl sulfate media. In 1% sodium lauryl sulfate media, the phenoldiphine release was 108.1% in 2 hours for formulation 17, containing an increased amount of lactose compared to 22.1% in formulation 16, containing a higher amount of microcrystalline cellulose. The obtained result agree with the previous experiment, confirming that use of lactose, its role is in enhancing the acceleration of dissolution rate. So if lactose is more, it will increase the dissolution rate of the tablet. Formulation 18 and 19, this formulation was formulated by differing the PVP amount, polyvinyl pyrrolidone amount. So in formulation 18, PVP was used 10 mg, and in formulation 19, it was used 20 mg. In this case, there is no significant difference. If we see the increasing the concentration of PVP in the formulation significantly enhance the in vitro release rate in the media, but no effect in CTAV media. In CTAV media, release is less than 80%, and in sodium lauryl sulfate media, the release is achieved to 100%. So this phenomenon is possibly due to an increased degree of hydrophilicity and PVP dissolving faster than the main matrix forming polymer. This by studying all these different 18 formulation, we achieved two final formulation formula. In this final formulation, we can see the release profile of our product as well as innovator product. Reference product is gray and our final formulation is blue. So the dissolution profile of blue and gray is overlapping, and if dissolution profile is matching with the innovator, that we are achieved with our target. If we conclude this, the present study reveals that some factor considerably modify the dissolution rate, which is particle size, proportion of the different excipients, type of polymers used, and amount of some excipients like lactose and polyvinyl pyrrolidone. So we optimize these different excipients in order to achieve the desired release profile, and the release profile was not significantly affected by microcrystalline cellulose or by compression force. If we change the compression force from 45 and 90 or 100, it will not affect the dissolution profile. In summary, in the dissolution study, several factors must be considered. Dissolution medium, due to different influence of used surfactant and the use of reference product in order to establish the correlation for the in vitro studies of experimental formulations. But this is only the one parameter which we study in this case, dissolution. We have to study the assay of the formulation. We have to study the content of impurities present in the formulation which we made. So that study also has to be done as a part of pre-formulation study. But since we are making the extended release formulation, the rate limiting factor was dissolution, and we need to study the profile of dissolution compared to the reference product. Thank you so much. Dr. Kalpana.
Yes, thank you so much, sir. I think you have covered it all round, starting from dissolution, the different factors, not only covering the aspect of the drug, but the dissolution media, how it is actually affecting, the presence of surfactant, right? So all that aspect, especially the students have been learned like what are similarity factors or dissimilarity factors, F1, F2, and how they are actually getting implemented in the case study, in the real example. So that was quite exciting to understand. And I think now I would request the students to please unmute themselves, or Ananya, I think they can do that, right? They can unmute themselves.
Yes, ma'am.
Turn on their cameras, introduce them first, and then they can ask questions or can share their feedback also, even if they don't have any questions, right? So can we start one by one? You can raise your hand. Priya, just introduce yourself and share your feedback. Can you someone else, right? You are not audible. Anyone else? Asha, can you unmute yourself?
Am I audible?
Yes, first introduce yourself.
Good afternoon, sir. Myself, Asta. And sir, as ma'am told us that you have worked.
Your voice is not clear.
Good afternoon, sir.
I think there is some network issue. So
Yeah.
can you try once more?
Good afternoon, sir.
Yeah, good afternoon.
Sir, am I audible?
Yeah, please.
Sir, as ma'am told us about your experience in the industry, can you please explain like what you have experienced?
Okay. I started my professional career like studies in 2001. I admitted in B.Pharm in 2001 in GJU Hisar, Guru Jambesh University, completed in 2005, followed by M.Pharm in 2007. So after completion of M.Pharm, I joined as a lecturer in JCD Sirsa, Department of Pharmaceutical Sciences. So after around 6 months, I got an opportunity from Ranbaxy Laboratories Limited. I moved to industry because my basic interest was in industry. Although I enjoyed my teaching experience also, in the short tenure, I got a lot of love from students. After joining industry, I started learning about the research, real research, which is going into the industry, basically in analytical research. So as a M.Pharm, I am pharmacologist, but I did not get any opportunity to work as a pharmacologist. And if we see while studying in M.Pharm and B.Pharm, we are much focusing on pharmaceutical analysis. So that was also quite interesting topic for me. So I moved to analytical industry. I got an opportunity in the analytical industry. So in 2007, I joined Ranbaxy. in analytical research. In analytical research, I worked on method developments initially, then followed by method validations, analytical methods which are required to assay the compound, which are required to make a dissolution profile, which are required to assess the impurities present in the drug formulation. So these are the analytical methods which we use to develop. After developing the method, these methods should be validated. So I got a rich experience in method validations also after development, and those validated methods which we transfer to the manufacturing locations. In manufacturing locations, we are formulating our commercial products. Those products are required to be tested according to the specifications, and all the tests listed in the specification we use to transfer to the methods for those tests we use to transfer to the formulation locations, manufacturing locations. So I have experience in method development, validation, method transfers, all these aspects of analytical research. Currently, I am working in Milan Laboratories. I moved here in 2020, located in Hyderabad. It is basically an API company. After merger with Upjohn Pfizer, after merger with Pfizer, it became Whitetrace. So you can see the symbol Whitetrace. This Whitetrace is a combination of Milan plus Pfizer, generic version of Pfizer. So two companies come together to make a different entity to give the safe medication to the patient. And it is one of the biggest generic company in the world. And here I am working in assessing the risk of genotoxic or carcinogenic impurities which are being generated into the formulation. Recent in 2020, nitrosamine came into the picture. This nitrosamine is nothing but the compound having nitroso group. Nitroso group compounds are much more prone to mutate our DNA to make the cancer. So in simple words, we can say if nitrosamine impurities are present in a formulation, it will cause cancer to the patient. So we have to monitor, assess the risk of presence of these impurities in our formulation. We have to reduce the level up to the ppb level, or if these impurities are not present in formulation, it will be very, very good for the patient safety. So we are contributing to provide the safe medication to the patient away from the cancer. We are using specialized technique for these determinations like LCMS, GCMS. LCMS is HPLC linked to mass spectrometry. In this, by using this technique, we can determine very, very trace level of the impurities present in the formulation. For these impurities, we have to develop new methods for different impurities in different type of formulations. We are developing methods, we are validating those methods, and we are transferring those methods to the manufacturing sites. And manufacturing sites are testing each and every drug product to ensure that these nitrosamine should not be present in the final formulation which are going to the patient, which is very, very important for the patient safety. So this is my overall profile up to this. Thank you so much for asking this question.
Thank you so much.
Thank you so much, sir. If there is some network issue at her end, I think, who else? Please raise your hand. Yeah, Varithro will be speaking next. Yes, Varithro, please continue. Are you able to unmute yourself? Ananya, can you help Oritra Sahab to unmute?
Yes, ma'am. I have given in the permission. Yeah.
Yes.
Hello, sir.
Hi.
Sir, my question is, what
First,
are the
introduce yourself. Sorry,
I am also Shah. Um, Sir, my question is, what are the skills we do require for to join the industry?
Sorry, your voice is very low.
so he's asking basically what skills he require to enter into pharmaceutical industry at a very beginning or initial stages.
Okay. So right now you are working in you are doing M.Pharm, right? First year.
Yes, sir.
Okay. So may I know your specialization?
Yes.
Your
Yes, sir.
specialization subjects?
Pharmaceutics. Sir.
Pharmaceutics. Great.
Yes, sir.
So if we are studying that pre-formulation studies, it is very helpful for pharmaceutical industry. So if you want to enter into the pharmaceutical industry, you should have all the basic knowledge which are related to academics, and you can have some references from the industry. You can be in touch with industry people so that they can provide your CVs and resume to the resource persons, and they can recruit you. They can offer you opportunity. They can interview you. But before that, you should have clear thoughts, clear objectives, what you want to do, where you want to achieve, in what area you want to work.
Sir, I also have another question. That what are the challenges a fresher may face after entering into an industry?
What are the challenges
A fresher may face after entering into an industry?
The
after entering
challenges
into
are
industry?
faced by a fresher once he enters into the industry, the pharmaceutical industry? What are the challenges? How to cope up with those challenges also?
So yeah, it is like very basic question and very important for every individual. So when you enter as a fresher in an industrial and in a corporate world, you need to be competitive, you need to be honest, and we face a lot of challenges when we enter. Like we are directly going from college to industry where we have to perform according to their expectations. So when we are fresher, we take some time to understand their expectations, to perform there. So that phase is very critical in order to assess your growth. Challenges: the culture and the climate is entirely different from college. That will be highly professional. So challenges will be very high. But that is not very difficult if person is full of knowledge, is open to learn, can explore the world. This is the base.
Thank you, sir.
Like see, I studied pharmacology in M.Pharm.
Yes.
And worked in analytical industry, and I am growing. So we need not to worry that what we are doing, but we need to know, we need to learn every day, we need to meet the company expectations. That is the prime requirement.
Right.
So I learned analytical skills as well.
Satisfied?
Thank you, sir. Thank you, ma'am.
Next question,
Welcome.
please. Yes, Surya.
Good afternoon, sir. I'm Surya Badrinath from M.Pharm first year pharmacy.
Hi.
You're muted.
One minute, hold on.
So
I need to take
the
a
session
one
was
call.
really
Sir,
insightful.
sir, please.
Hi. Thank you for giving me time.
One thing, can you please unshare your screen once? We want to take a group photograph. Everyone, please turn on your cameras if possible. Ananya, can you please do the favor?
Sure, ma'am.
I think it would be nice if we can show the class.
Yes, for sure.
I see that at many places, the entire class is attending this session. This is really great, but we are not able to see in the camera. So I request everyone, please, if possible, you arrange your camera in such a way that we will be able to see the entire class students.
Along with the teacher.
Yeah. Well, students are more important than teachers.
Yes. I thought I should not miss myself in this photograph. That's why.
Yeah, no, that's true. So I think Sudeepa Singh, I can see many. Medha, I think Medha. You show your entire class. Jayasree. If you can show your entire class, would be great. Aditi, also I can see students behind you. Okay, I think is much better.
Surya?
The group photographs have been taken.
Click.
Yes, ma'am.
Thank you, Ananya.
Okay. Yeah, let's continue the discussion.
Yeah. Thanks, Rajan. Yeah. So, Surya, you were asking question. Please continue with that.
Yes, sir. So my doubt was, what are the software side opportunities for pharmaceutical industry, like forecasting, etc.?
Software opportunities.
Yes, sir.
Very good question. Actually, automation is very, very important, and in every pharmaceutical industry, we are using lot of softwares. So I don't know what kind of softwares you want to ask about, but as a pharma industry, we are reliant on many softwares, starting from archival of documents to the application part. So we are running every instrument by using software. So we are completely dependent on softwares. Each and every instrument has different software.
My doubt was like not to the industries, like desk job, like for business forecasting, market analysis. What are the jobs like that?
Market research.
Market research.
Sorry, I do not have this experience, so I cannot comment much more on this area.
No problem, sir. Thank you.
Yeah. Anyone else? Yes, Jasmine will continue. Yeah.
Good morning, sir.
Yeah, good morning.
So myself, Jasmine. I'm a student of MPharm first year, Sutix department. Since I have joined my BPharm in 2018, from that time, I have come in a lot of experiences where people say that industry is not for women. I just wanted to ask you, does this notion still continues? And if it continues, like I can show you 80% of my class comprises of girls. So if this notion continues, please give us some measures to break this notion. Like what are the steps we can take?
Actually, your question is very, very relevant. In pharmaceutical industry, there are a lot of departments, right? So
yes, sir.
In few of the departments, women are not being hired much because lot of exertion is required, lot of late stayings and night duties are required. So in those cases, people avoid hiring women, but it is not like that. In those departments also, more than 20% women are there. Like in analytical research, we in this also more than 25% women are in our department. But if you see regulatory affairs, intellectual property rights, various documentation related to analytical research, corporate quality assurance, in those departments, more than 60% women are there. So it is not like there is less scope for women. But while selecting the department, we should be cautious that in which department we can get more opportunity. So for that we need to speak to the industry people, which department should be will be suitable, which department has lot of scopes.
Great.
Okay, sir.
Hope you are
Thank you, sir.
Who's
Yes,
next?
sir.
Anu?
Hello, sir.
Hi.
It was indeed a very insightful presentation on pre-formulation studies today. And we actually learned a lot more than our basic academic endeavors. Also, I would like to ask you about how pre-formulation in industry is different from the laboratory levels that we do in our colleges.
Very good question. Very good question. Industry is the bigger picture of academics. So in academics, we used to formulate a formulation by low-grade instruments. Even in academics also, we are much explored. We are using recent equipments also. But industry is real application, which is broader picture of academics. So it is not very different. Pre-formulation starts from basic. It starts from academics also. So while I was presenting, if I was a student 15 years back, I could present in much more better way than today. So academics comparison between academics and industry is just simple. It is the bigger application of the academics.
Okay, sir. Thank you, sir.
Anyone else? Medha?
Good afternoon, sir. Myself, Megha. I'm actually from Farm Pharmaceutics. Sir, I want to ask you,
Please continue.
sir, how I want to ask you how to get more exposure in research and development.
Sudeepa, can you please continue with your question?
Ma'am, am I audible?
Yes.
Good afternoon, sir. Firstly, I would like to extend my sincere thanks to you for such a great presentation and information that you have given to us. So I would like to ask, as I am from a pharmaceutics background, like I have done my master's in, like I am doing my master's in pharmaceutics. So being a pharmaceutics specialized person, what are the job profiles that I can get in industry?
Yeah. As a pharmaceutics, you are a formulator. So first preference will be the formulator, as a formulator. So there is every pharmaceutical industry which require formulator. So lot of scope is there, right? Apart from formulation, you can go to regulatory agencies, regulatory department also. As you are aware of different kind of formulations, you can learn more about the regulatory aspects also.
Okay, sir. Thank you so much.
Sarika want to ask a question? Yes.
Very good afternoon, sir. Am I audible?
Yeah, good afternoon.
Good afternoon, sir. My name is Sarika Gupta. I'm from M-Pharm Pharmaceutics first semester. So my question is like, what piece of advice would you like to give us since we are a first year master's students? So I would like to hear from you what we need to do, focus more in this 2 years period.
Very good question. Actually, while we are studying, we need some direction. So this is question regarding that only. When you are studying, you should be open for learning. Don't think that you are you are doing pharmaceutics. You can learn every aspect of the pharmacy. You can grab more and more knowledge, but you should aim for something. So you should know what are the different departments available in pharmaceutical industry. Where do you want to go? Where do you want to go in academics? So that is also open. academics is also very good for person to go, because you can share your knowledge, you can learn more, you can be in touch with academics, you can do PhD there. In pharmaceutical industry, we have different departments, so we should know what is the function of what department, and according to those functions, we should select in which department do you want to go. According to that, you should learn what is the requirement to enter into that department. So you can make an aim that whether you want to go in academics, you want to go in industry, in which department you want to go, and you should be open to learn.
Thank you, sir. That was very helpful.
Thank you. Who else? Rimjin?
Good afternoon, sir.
Yeah,
I am
good
Rimjim from MPharm first year, Pharmaceutics Department. Am I audible?
Yeah.
Yes, sir. So I wanted to ask if we talk about Pharmacovigilance Department and Quality Assurance Department, so between both of them, which department has more scope?
Pharmacovigilance and
Quality Assurance.
Quality Assurance. Okay, so these are entirely two different departments.
Yes, sir.
In pharmacovigilance, there should be knowledge of pharmacology also. In corporate quality assurance, that is not related to research. So that is related to maintain the quality in the pharmaceutical system.
Yes.
Quality and compliance. So for quality and compliance, if you want to go in that area, that is also have a good scope. Every pharmaceutical company has this department, and strength of this department is also huge. So scope is more in this case. Regarding pharmacovigilance, I am not aware much about that department.
Yes.
The function of
QA
corporate quality assurance.
Thank you so much, sir.
Yes. Next, Sakshi.
Good afternoon, sir.
Yeah,
Sir,
good
this is
afternoon.
Sakshi. I'm doing my MPharm in pharmaceutics. Sir, I have a basic question. Like when we all are here for jobs, so what are the additional skills we need to put on in our resume so that it is different from others and we can get the job easily?
Sorry, please repeat your question. Kalpana, can you?
Yes, she is actually interested in the skills she should acquire so that she'll easily get the job as per industry. What additionally she should do as of now so that she'll get better opportunity?
Actually, basic knowledge and strong base is required to enter into industry. Basic knowledge in your subject.
Sir, apart from academic skills.
Apart from academics, yes, you should know industrial application during studying in academics. Means what you are doing, you try to correlate what is happening in the industry. What is the industrial expectations? So if you know some person working in industry, you can be in touch with them, what they are doing, what you can do after entering to the industry. So if you communicate more with different type of people working in industry, you can learn what is happening in the industry. But strong base is required. We think as we are studying B Farm or M Farm, we should study all the subjects well. Every subject has its own importance. One day, 2 day studies will never give fruits. Every day study will give fruit. Because everyday study will make your base strong. Your everyday study will improve your concepts. So developing concepts is much more required. That will be applied.
Yes, sir. Thank you, sir.
We are satisfied. Want to ask? Aditi? Oh, yeah. Hope we are not taking much of your time.
No issues.
Okay.
Good afternoon, sir.
Good afternoon.
I am Aditi Sinha from Pharmaceutics Department. Sir, I want to ask, if I want to go in Research and Development Department, so will I require to gain some experience in industry before going to that? Will be it beneficial?
Actually, Research and Development Department नहीं होता है, मतलब रिसर्च एंड डेवलपमेंट इंडस्ट्री है, उसके अंदर डिफरेंट डिपार्टमेंट है. रिसर्च एंड डेवलपमेंट इंडस्ट्री में प्रोडक्ट डेवलपमेंट डिपार्टमेंट होता है, एनालिटिकल रिसर्च डिपार्टमेंट होता है, केमिकल रिसर्च डिपार्टमेंट होता है. सो अगर आपको रिसर्च में जाना है, स्पेशली as a pharmaceutics, you need to think about the product development department, because you can apply your pharmaceutics knowledge there much more better way. So in product development research, these pre-formulation studies are being done. For every formulator, they have to study every aspect of the formulation, starting from active pharmaceutical ingredient to various excipients and their interactions also. So in order to make a good safe to develop a good safe medication for the patient, you need to learn each and every aspect in your studies, and try to be in touch with industry people if you want to go there, that what they are doing different from academics. So you will be aware that what you can improve in yourself, what skills you need to develop.
You are satisfied?
Okay, sir. Thank you. Yes, sir. Thank you, sir.
Okay. Arushi?
Good afternoon, sir.
Yeah, good afternoon.
Myself, Arushi. I am from M-Pharm Pharmaceutical Department. So I want to ask, so what are some special specifications that we must have before entering the industry?
As I already said that your base should be strong. You should know where you want to, in which department do you want to go in industry, what is the requirement, and your basic knowledge is very important. What you are studying should have more applications. You study,
sir, in terms of instruments.
In terms of instruments. So in which college you are studying, if high grade instruments are available there, you can learn each and every instrument available in your department. Apart from that, you can reach out to various other research areas. They can also support, like DRDO. You can reach out those people. You can go to industries for some trainings as a part of your curriculum, or after completion of your M-Pharm first year, you can go to industry to perform project in the industry. So if in 1 year you work there, you will be aware what is happening in the industry, and you can own yourself in that area.
Okay, sir. Thank you.
Jayashree?
Good afternoon, sir. Myself Jayashree from M-Pharm Pharmaceutics. So my question is, what are the job opportunities after completing M-Pharm in abroad?
In abroad?
Yes, sir.
Very difficult. Actually, in abroad, various pharmaceutical companies are there, but their requirement is like work visa. So if you have the work visa, you can easily get the job. But company from other countries will not accept any application unless otherwise you have work permit. So
Okay,
if you want to go abroad, you can go there as a student to perform some higher studies and get job there, and after getting job you can get the work visa easily.
Okay, thank you, sir.
Meghna?
Yes, okay.
You're audible. Please continue. Is your voice audible in the meeting? No. Can you be a bit louder or unmute yourself properly? Take from your classmate if possible.
Very good afternoon, sir. Myself, Meghna Dhi, and currently I'm pursuing M-Pharm Pharmaceutics. I'm a first-year student from MIT University, Noida. So my question to you was, like while sitting for interviews, what qualities does the company look for us in? Like, I'm not asking like a role-specific question, like role-specific qualities, but like in general, what qualities I should be having in myself that would put me ahead, less than my peers. So
You should be confident, you should be knowledgeable. So every industry hiring from colleges, they will look the confidence in the candidate, whether they can work under extreme conditions or not. And skills are much more required. So skills means in which department you are applying, you should know the basics at least.
okay.
Discipline also.
Okay.
Yes, the girl in the last.
Thank you so much, sir.
Good afternoon, sir.
Yeah, good afternoon.
I am Mayuri Goyal from M-Pharm first year Pharmaceutics department. So I want to know about what were the challenges industry faced during the pandemic situation.
Great question. Great question. So if we think about the pandemic situation, it was very, very, very difficult for the industry people, because we need to continue develop the medicines. We need to work every day. Other industries can shut down their industries for some times, but pharma industry cannot shut down, because medicine should be available to the patient every time. And in pandemic situations, lot of medicines were required for the patient during hospitalizations also. So in those cases, in those times, we managed to take various protective equipments with us and work continuously. Sometimes we worked in shifts, because if 100 people are working in one department, so that will be more crowdier. So 30% worked in A shift, 30% worked in B shift, 40% worked in general shift, like that. But work was on. In pandemic situation, work was on for pharmaceutical industry. I hope this was your question.
Yeah. Thank you, sir.
Any other question? Online attendees, Mr. Lokesh, or if you want to unmute Himanshu Dagar, Lokesh, Harsh. Okay. Sir, I have a question related to pre-formulation studies.
Very good. So this was our subject.
Yes, this was our subject. Other than that, everything was asked. But I think as a teacher, I mean, I had doubts in explaining to them sometimes, like extended release, prolonged release, then immediate release. From immediate to prolonged, we can of course differentiate between them and explain them well. In controlled, prolonged, and extended, so a clear-cut explanation to them so that they can understand the concept in a better way from an industrial expert perspective.
Okay. So this is not like industrial aspect. This is like simple terminologies.
Yes.
So if we talk about immediate release formulation, these are the formulation which release immediately after administration, like within 30 minutes. If we talk about the delayed release formulations are usually enteric coated formulations, in which we delay the release for some time, and after delaying for some time, it will release. For example, enteric coated tablets. So those formulations which are very much prone to react with acid, those should be coated with enteric coating, that it will pass through our stomach. It will not interact with the acid present in the stomach, and after 2 hours, it will pass into the small intestine and release there. So for some time, for some 2 hours, we delayed the release of that formulation. That is delayed release formulation. If we talk about the controlled release formulation, means we want to control the release of formulation. So in that case, all these sustained release, modified release, and controlled release, all these are similar. Means we are controlling the release of a formulation based on our expectation. If we want to formulate an extended release formulation, we want that drug will enter into the body and slowly release the drug with time. Like in 2 hours, it should release 10%. In 4 hours, it should release 40%. In 8 hours, it should release 70%. And in 12 hours, it should release 100%. To get the continuous response from the medicine, pharmacological action from the medicine, that formulation is designed. Means if you want to take a medicine 3 times a day, so immediate release formulation can be taken. If you want to take medicine not 3 times, only 1 time a day, in that case, extended release formulation will be very helpful, right?
Right. Fine. And once we do that for especially like nano drug delivery systems, we generally carry out dissolution studies on them. So ideally, after how much time, because they are not getting released up to 100% for prolonged duration, even for we have done it for like 24 hours or beyond that. So ideally, up to what time that dissolution profile should be compared for them, like especially the nano drug delivery systems and all.
Okay, so the dissolution profile for nano drug formulations, like 100% release should be there. So if there are some specifications that up to 12 hours it should be more than 85. So if you achieve 86 or 88 also, it will be good.
Okay.
So we need to meet the specifications as per regulatory
expectations, right? Fine. And one more thing, what I actually look forward, it's a general question, like we are developing nano drug delivery system from the past decade, right? But if I see the market, there rarely I find very few formulations which are being marketed. So according to you, what are the reasons for that, and how more and more nano drug delivery systems can be in the market?
Yeah, it is very good question. Actually, in market, we use to find solid oral dosage forms, mostly.
Yes.
Solid orals. So these are more convenient for the patients, right?
Yes, yes.
So if we are developing nano formulations, industrial application is very less. Industries are not started making these formulations much. So few departments are working in these areas also. I hope in future we can see some more formulations in the market. These formulations are costly also.
Yes, actually. Okay, so I think if there is no more questions, we are near the end of the session. So respected Dr. Rajendra Dhaka, Associate Professor, IIT Delhi, Chair INYAS, Dr. Sandeep Arora, Director, Amity Institute of Pharmacy, and our most valued speaker, Mr. Vaibhav Kumar, all INYAS members, and especially NCR chapter INYAS member, and all the audiences who joined throughout the country. It's my privilege to have been asked to propose a vote of thanks on this occasion. So any lecture cannot be a success without a learned speaker. I, on behalf of NCR chapter of INYAS and Amity Institute of Pharmacy, the entire organizing team, and on my own behalf, extend a very hearty vote of thanks to the speaker of today's session, Mr. Vaibhav Kumar, for accepting my request, gracing the occasion, and sharing with us his words of wisdom and opinions today. I would like to take this opportunity to place on record our hearty thanks to Dr. Dhaka for the perfect guidance and encouragement as he has extended to all of us. I must mention a deep sense of appreciation to Professor Sandeep Arora, sir, for always motivating us to get industrial experts' input in the existing curriculum, and for his prompt response for approving this wonderful talk. I may like to express the sincere thanks to Dr. Veda Krishnan also, who is coordinator of NCR chapter, for her continuous efforts to make it possible. I also extend my thanks to INYAS office, especially Ananya, and AIP office, especially Mr. Mahesh, for his enormous cooperation for the technical support for making this event happening. I also wish to thank our audiences for their patience, their enthusiasm, and the positive participation, along with asking questions and making it more interactive. Thank you so much.
Yeah, thank you for giving me opportunity to speak in this platform.
Our pleasure, sir. We'll invite you multiple times now.
Okay, I will be happy to participate.
Thank you. Thank you so much. Can we close the meeting now?
Yeah, thank you.
Thank you. Ananya?
Yes, ma'am. I'm ending the meeting for now. Thank you.
Bye, everyone.
Bye, everyone.